Hey, how are we doing everyone? Welcome to the Bearded Medic. In this video we're going to start to look at the humoral response, which is the response mediated by B cells and antibodies. We're going to go through their activation and how they start to produce antibodies specific to the pathogen, and we'll also look through the main actions of some of those antibodies. B cells can either be activated with or without T cells. There are two methods by which B cells can be activated without the need of T cells. Our bodies have naive B cells in circulation, and these B cells can come into contact with foreign pathogen. The B cell binds via their B cell receptor, which is a membrane-bound antibody, usually IgD or IgM. This causes a signal to be sent in the cell. They also bind to pathogen with their toll-like receptors, which sends another signal. So binding from both receptors triggers the B cell to turn into a plasma cell and automatically release antibodies to neutralize that pathogen. Now these antibodies don't have the highest affinity, but the hope is that they can make the pathogen ineffectual. Usually the antibody IgM is released, and it's released as a pentamer, so it agglutinates the pathogen and stops them binding to other cells, but it can also recruit complement. B cells can also be activated by macrophages and dendritic cells after it recognizes a foreign pathogen. The other antigen presenting cells can release B cell activating factor. This causes the B cell to release class-switched antibodies, usually IgG or IgA. IgA is released as a dimer. Now for how T cells activate B cells. They do this in the lymph node. Antigen from pathogens can flow to the lymph node via the lymphatic system and can be picked up by phagocytic receptors of follicular B cells, as most naive B cells are found in the primary lymphoid follicle of the lymph node. They phagocytose this antigen and present it on MHC2. And as it's foreign, our B7 co-stimulatory molecule is also upregulated. They then migrate to the T cell zone or paracortex to be activated. When going through T cell activation, we mentioned the T follicular helper cells, and their role was to activate B cells. And remember, this T cell was activated by a specific antigen that has been presented by dendritic cells. If this same antigen is being presented by the B cell, the T follicular helper cell will bind, and a similar activation process the CD4 cells went through, the B cells will go through. Activation, survival, and differentiation. But the differentiation process in this case is the T cell releasing cytokines, and these cytokines will dictate the class of the antibody produced for this pathogen. Now the B cell needs to get to work on creating these antibodies tailored to the pathogen. So when the B cell is activated, it moves away from the T and B cell border back into the primary lymphoid follicle, and then replicates to form what's called a germinal center. This can take up to a week after the B cell has been activated. The germinal center has a dark zone and light zone, and the dark zone is where B cells, now called centroblasts, replicate. What happens during this replication period is important. In order to create a specific high affinity antibody, the centroblast undergoes two processes while replicating. They are somatic hypermutation and class switching. Somatic hypermutation changes the B cell receptor's variable region. After each replication, the centroblast is mutating and proliferating, producing a slightly different receptor each time. The aim is to increase its affinity to the pathogen. Also, they express the chemokine receptor CXCR5. Follicular T cells constantly co-stimulate with centroblasts in order to maintain their survival. We also have follicular dendritic cells, which are constantly checking these B cells for the quality of the antibodies produced. And these interactions usually occur in the light zone of the germinal center, as the CXCR5 on centroblasts are attracted to CXCL13 released by the follicular cells. Only the best centroblasts go on to become centrocytes, as they have antibodies that have extremely high affinity to the epitope, or the part of the pathogen they will bind to. Those with low affinity are apoptosed by what are called tingible body macrophages. Now we need to make different classes of these high affinity antibodies, so the pathogen can be destroyed in all different manners. Class switching, as the name suggests, changes the class of the antibody, like if it's IgA, IgG, IgE, IgM, or IgD. This, as we mentioned, is dictated by the cytokine the T follicular helper cell releases. This will turn the centrocyte into either a memory B cell or a plasma B cell. The plasma B cells are the ones that will secrete different classes of the antibodies. The main cytokines to note are interferon gamma, IL-4, and transforming growth factor beta. Interferon gamma leads to IgG antibody subtypes Ig3A and Ig2A. Think gamma and IgG. With IL-4, IgE and IgG1 are produced. For this, remember that IL-4 also increases Th2 responses which sensitize eosinophils. So IL-4 eosinophils. With transforming growth factor beta, IgA is the main antibody produced, and you can remember this by remembering that TGF-beta also produces T regulatory cells, which lead to an anti-inflammatory response. And anti-inflammatory responses are mainly desired in the GI tract, where IgA is mainly released. So somatic hypermutation and class switching usually occurs when B cells replicate in the germinal center. The constant tweaking eventually creates antibodies that have an extremely high affinity to the epitope. Remember, epitope is the part of the pathogen that antibody binds to. What happens next is dependent on the class of those antibodies, so here's a brief overview of the main action of some of the antibodies. IgG has multiple actions. It can sensitize natural killer cells to virally infected cells by binding on the viral epitope, opsonizing that cell. The natural killer cell can then bind to the antibody's Fc region, like an anchor, then does its job via perforin and granzymes. IgG can also directly bind to the pathogen at the site of infection, which opsonizes the pathogen again, but this time to phagocytes. These phagocytes bind using their Fc receptors. This induces phagocytosis of that pathogen. IgG and IgM also activate the classical pathway of the complement system. They bind to the pathogen and recruit the C1q complement protein, which binds to the antibody's constant region, eventually leading to C3b recruitment and phagocytosis. As we've said before, IgE should trigger you to think eosinophils. It binds to parasites which sensitize eosinophils to the area. Eosinophils bind to IgE via their Fc epsilon receptor 1, and eosinophils release their granules to destroy a parasite. Mast cells also have this Fc epsilon receptor 1. IgE binds to these receptors, and if antigen then binds, these receptors cross-link and the mast cell degranulates. This releases histamines, leukotrienes, and various other enzymes and cytokines, and this is particularly seen in allergies. Finally, as we've mentioned before, IgA can be released across epithelial barriers to neutralize pathogen and stop them binding to other cells. IgG and IgM can also neutralize but can't pass through barriers. So let's review what we've covered in this video. B cells are antigen presenting cells along with dendritic cells and macrophages. They can be activated to secrete antibodies without T cells by direct recognition or other antigen presenting cells releasing B cell activating factor. They then secrete relatively low affinity antibodies to neutralize the pathogen. B cells can also be activated by T cells in the lymph node, where the B cell then moves back to the B cell zone and replicates to form a germinal center, undergoing somatic hypermutation and class switching to create extremely high affinity antibodies. Only the B cells that create these high affinity antibodies become memory cells or antibody secreting plasma cells. Each class of antibody has a different function. IgG goes to opsonize infected cells for natural killer cells or pathogens for phagocytes. IgG and IgM can activate the classical pathway of complement, again ending in opsonization and phagocytosis. IgG, IgA and IgM are secreted to neutralize pathogens. And finally, IgE sensitizes eosinophils to parasites and also causes mast cell degranulation. So this marks the end of the video. If you have any questions, be sure to leave them in the comments. If the video was helpful, please leave a like and subscribe for more. Thank you very much for watching, and see you in the next video.